Hi everyone. Welcome to the session on biochemistry, and this presentation will see on nucleic acids. When we look into the study of nucleic acids, the history, you can see that it was in 1869 the DNA was discovered by Johann Friedrich Miescher. He isolated DNA from the salmon sperm, and it was named as nucleic acid because it was found that the particular biomolecule, it was obtained from nucleus, and it was acidic in nature. So, putting both the concepts together, the term nucleic acid was given to the biomolecule. And later in 1944, Avery, MacLeod, and McCarthy, they proved that DNA was a genetic material, or we can say DNA contained the genetic information. Later in 1953, it was James Watson and Francis Crick, who came up with the double helical model for DNA. And they came up with a structure based upon the studies, X-ray diffraction studies conducted by Rosalind Franklin. In 1968, Nirenberg and many other scientists, including Hargobind Khorana, they decoded the genetic codes. And later in 1981, Gilbert, Sanger, they came up with the DNA sequencing method, which could later be employed for the genomic studies. Okay, so this is the basic, we can say, the timeline of discovery and as well as various studies conducted on nucleic acids. Now we'll see about the general aspects of nucleic acids here. When you look into the nucleic acids, we can say that all living organisms with a nucleus or without a nucleus do have nucleic acid in them, except for like prions. Prions, they don't have any nucleic acid. Instead, they have only proteins, and these proteins, they are infectious in nature, and they do have the property of replicability. But coming to other organisms, all other organisms do have nucleic acid in them. In the case of eukaryotes, that is those organisms having a well-defined nucleus, in them more than 98% of cellular DNA is present in the form of double-stranded linear DNA. Okay, so obviously DNA, they are found in two forms. It's linear and circular. Okay, so in the case of eukaryotes, more than 98% of DNA present, or nucleic acid, especially the DNA present in the cell, it is in the form of double-stranded linear DNA. And all the eukaryotic DNA, it is found complexed with proteins which are known as histones. Unlike the eukaryotic DNA, in the case of prokaryotes, in the case of eukaryotic mitochondrial and chloroplast DNA, so they have circular DNA. And these circular DNA, they are never found complexed with histones. So the mitochondrial DNA as well as the chloroplast DNA, they are almost similar to a prokaryotic DNA, which is circular in nature and not being complexed with histone proteins. Now, that was about the DNA. When we speak about other nucleic acids, we have plenty other nucleic acids, and out of that, the most important are RNA, the different types of RNAs. Okay. The RNA, the ribonucleic acid, they are found in the cytoplasm of both prokaryotes and eukaryotes, and they are found in association with non-histone proteins, or they may not be. Okay, they may be found free in the what you call the cytoplasm. Okay, so DNA, it may be either confined within the nucleus, as in the case of eukaryotes, or it may be found inside the mitochondria or chloroplast, again in the case of eukaryotes and specifically in the case of plants, respectively. And in the case of prokaryotes, if it is present, it may be in the nucleoid. Okay, so this is about the nature of nucleic acids found in different types of cells. Coming to the structure of nucleic acids. Nucleic acids, they are polymers of nucleotides, just like proteins being what they call polymers of amino acids. The nucleic acids, they are polymers of nucleotides, and these monomeric units, that is nucleotides, they are bound by 3' 5' phosphate bridges, the structure of which we will be looking in detail in the coming presentations. Okay, so what is a nucleotide composed of? As we have already seen, nucleic acid, it is a polymer of numerous nucleotides held together one after the other in a sequential manner, right? So we can see how a nucleotide is what a nucleotide is composed of. A nucleotide, if we look into, it is composed of three major molecules: a nitrogenous base, a pentose sugar, and a phosphate molecule. A nitrogenous base, that is a molecule which contains nitrogen. Obviously, that is a name, isn't it? And there are two types. Nitrogenous bases can be classified into two types based upon its structure, that is purines and pyrimidines. The structure and other details we will be looking in detail in the coming slides. Then the second one is pentose sugar. Pentose sugar, that is a monosaccharide, which we have already come across in one of the previous presentations. The pentose sugar, it is a 5-carbon monosaccharide, and the major pentose sugar found in nucleic acids, whether it is DNA or RNA, it is the ribose or deoxyribose. Okay, deoxyribose in the case of DNA and ribose in the case of RNA. Then we have is a phosphate molecule. Phosphate molecule is H3PO4, where a phosphorus central atom, it is being connected or bound to 4 groups, of which 3 hydroxyl groups. It is attached to the phosphorus by way of single bond or sigma bond, okay, and 1 oxygen bound by double bond. So that is how the H3PO4 is arranged. So a nucleotide, it is composed of 3 major molecules: a nitrogenous base, a pentose sugar, and a phosphate molecule. Okay, so this is the structure you can see here. So here, this is the pentose sugar. Okay, so you can see here it is in the ring form, and the pentose sugar, it is attached to a nitrogenous base and a phosphate molecule. This is a phosphate molecule. And in a nucleotide, the nitrogenous base is attached to the first carbon on the pentose sugar. Okay, the pentose sugar, they have 5 carbons, and the first carbon of this, it is attached to the nitrogenous base, while phosphate is attached to the 5th carbon of the ribose sugar. So this is how a nucleotide is being formed, right? Now, nitrogenous base, we'll be looking in detail in the coming slide. There are two types based upon the chemical structure. We have the pyrimidines and we have the purines, right? As we can see, pyrimidines, it is composed of a single ring, while purines, they are formed of 2 rings bound together. Now, sugars, they are of 2 types: ribose and deoxyribose. Ribose in the case of RNA, and deoxyribose in the case of deoxyribonucleic acid. What is the difference? The difference is in the group attached to the second carbon on the sugar. Here you can see in the case of ribose, the second carbon do have hydrogen and hydroxyl group, while in DNA we have 2 hydrogen atoms being attached to the second carbon. That is how it is deoxy, because ribose, from the ribose, 1 oxygen is removed from the second carbon position. Carbon in the second position. So that is how 2-deoxyribose. That is how the name has come. Now, we'll see in detail the structure of nitrogenous base. Nitrogenous base, it can be considered as a heterocyclic compound. Heterocyclic, why? Because in this, it is a cyclic structure where it is the skeleton, it is composed of carbon and nitrogen. So there is some other atom occupying the positions in the main the skeleton. Okay, the cyclic skeleton. That is a heterocyclic compound. And here it is a nitrogen containing compound. So it is a heterocyclic nitrogen containing organic compound. Okay, 2 kinds are there: purines and pyrimidines, which we have already mentioned. Purine, it contains 2 rings, one 6-membered ring bound to a 5-membered ring. Okay, so these 2 together forms the purine. In the case of pyrimidine, it is just a 6-membered ring. Okay, it contains only a ring made up of 6 members. Okay, so you can see here, this is how the structure is. Okay, you can learn the numbering. It is 1, 2, 3, 4, 5, 6, and then 7, 8, 9. You just need to know that the first numbering starts with nitrogen. Okay, and it goes in this way, right? But in the case of pyrimidine, again, it starts with a nitrogen, but it is just the reverse. Okay, here it is in the anti-clockwise, and here it is in the clockwise order. 1, 2, 3, 4, 5, 6. In both the cases, we can see the first and the third position is occupied by the nitrogen of the 6-membered ring. In the 5-membered ring, it is 7th and 9th position. You have to learn the structure of purine and pyrimidine. Now, we have different types of pyrimidids and different types of purines. We have 3 types of pyrimidids. We have the cytosine, uracil, and thymine, and purine, guanine, and adenine. Okay, you can say it is CUT and AG, right? Now, the basic structure of pyrimidine is this one, where the first and the third position is occupied by nitrogen, and the rest of the the positions are occupied by carbon. Okay, and every carbon, it is attached to a hydrogen atom, and there is alternate double bonds in the pyrimidine ring, right? So when we speak about this one, the different types of pyrimidine, the cytosine, uracil, and thymine, they are just modifications of the basic pyrimidine ring. You can see here, in the case of cytosine, it is oxy, double bond O at second position, and amine attached to the fourth position or the fourth carbon. That is how it is modified. Okay, and what about uracil? It is C double bond O, the second position, and C double bond O at the fourth position as well. Okay, and thymine, it is a modification of uracil. What is the difference? The second and the fourth carbon with double bond O, with along with that fifth carbon is attached to a methyl group. Okay, so we can say it is 5-methyl uracil. Thymine is 5-methyl uracil. Okay, similarly, I suppose we can see here, right? Okay, so this is pyrimidine. You can see it is a monocyclic molecule. It is a benzene type ring with only a 6 member in the ring, but first and third position being occupied by a nitrogen. Okay, here we can see it is the cytosine. It is 4-amino pyrimidine 2-on. In the sense, fourth position it is occupied by amine group, and second position it is having a C double bond O, ketone nature. Okay, so cytosine is 4-amino pyrimidine 2-on. Then uracil, it is pyrimidine 2,4-dione. In the sense, the second and the fourth position is having keto, that is C double bond O group. Okay, now thymine is 5-methyl uracil. They have 2 keto groups at second and fourth position respectively. Along with that, the fifth position they have a methyl group. I hope that is clear. Okay, now coming to the bonding. As we have already mentioned, the you can see here, this is how it is, isn't it? That is, the nitrogenous base, it binds with the first carbon on the sugar, isn't it? So in the case of pyrimidine, the first N1, this one, okay, the N1, it forms a link with the first carbon on the sugar, and since it is a sugar which is making up the bond is known as a glycosidic bond. Okay, so that glycosidic bond, it is through the glycosidic bond. So pyrimidine, it forms a glycosidic bond by way of N1 of the pyrimidine linking to the first carbon of the sugar. Okay, and the base linked to sugar, it produces what is known as a nucleoside. And when the base is a pyrimidine, the nucleoside is known as a pyrimidine nucleoside. Coming to the next group, nitrogenous base, purines. Purines, they are dicyclic molecules, and it is formed by the fusion of a benzenoid ring and an imidazole ring. Okay, the 6-membered ring is known as a benzenoid ring, and this 5-membered ring with nitrogen compound is the imidazole ring. Okay, now there are 2 types of purines, adenine and guanine. If you see adenine, it is 6-amino purine. So purine, you can see the structure over here. Okay, adenine, it is 6-amino purine. That is, in the purine, on the purine, the 6th position is occupied by a amine. Okay, now the amine is attached to the 6th carbon in the purine. And what about guanine? Guanine is, you can see here, the 2-amino, 6th carbon is in keto form. Okay, so 2-amino purine 6-on. That is guanine. I hope it is clear. Okay, so purine also binds with the first carbon of the sugar through glycosidic linkage, and there, instead of the first nitrogen, it is the 9th nitrogen. Okay, nitrogen in the ninth position of the purine binds with the first carbon on the what do you call the pentose